Well, hello, everyone. Thank you for coming to my presentation. So today I will be presenting on my internship project where I analyze for differentially expressed genes present in patients with diverticulitis. So as Marielle already said, for my internship, I worked with the Bioinformatics and Analytics Research Collaborative, or BARC. And this organization is a multidisciplinary center that aims to provide bioinformatics expertise and analysis to researchers across the university. They also do some work with external customers as well. And all of their work is dedicated to helping researchers analyze high throughput data. My internship started on May 1st and it ended on October 20th of this year. My deliverables included volcano plots, a gene overlap file, a gene count file, and also multiple cluster plots. So let's get into the actual process of analyzing for differentially expressed genes in diverticulitis patients, and also a little bit of a background on what diverticulitis actually is. So diverticulitis is the infection or inflammation of pouches that can form in the intestines. And you can see here with this image, I'm not sure if you can see my cursor, but if you look at the image, you can see these little pouches here in the large intestines, these little bulbous masses. So these are called diverticula or tics. And you'll hear me say tics throughout the presentation. So when I say tics, that is what I'm referring to. And so this image provides a visualization of tics present in the large intestine. And also here you can see diverticulitis present. So this infection or this inflammation that's going on right here. The rate of diverticulitis increases with age, and it is more common in individuals over 40. However, in recent years, there have been more patients below the age of 40 being hospitalized. So you can't discount a potential diverticulitis diagnosis in people below 40. A lot of people do not have any symptoms of diverticulitis, but those who do experience symptoms may have nausea, vomiting, fevers, chills, etc. Diverticulitis can also be severe and result in infection or rupture of the bowel, which you can see here in this image where we've got diverticulitis with rupture. Other complications can include abscesses, fistulas, and scarring. Despite all of this and how frequently we see it, it's really interesting that experts are actually not sure what causes this condition. There are some risk factors, which I included here, and those include genetic factors, obesity, lack of exercise, eating red meat and lots of fat, a lack of fiber, and there are actually a couple of more, such as the use of certain steroids. But we know these risk factors, but we don't know exactly what causes this and what contributes to the presence of tics in the large intestines in the first place, which gets a little bit more into my actual project. So throughout this internship with BARC, I analyzed gene expression levels from sequencing results from colon tissue samples collected from over 500 participants. A metadata file of these participants was also provided, and this included demographic and other clinical information. And also I should just say that my lights are flickering in and out. I'm not sure if I will lose power, but if I disappear, just know that that is why. But going back to the subject, this metadata file included demographic and clinical information like BMI, race, age, gender, and also the number of tics, remembering that tics are diverticuli. So how many are there, and also where are they located in the intestines? So my workflow is actually very similar to the sample workflow I have here. To the side of this slide, you can see it starts off with the actual biological samples, which in this case would be those 500 total RNA samples from colon tissue. These samples were prepared into libraries, pooled, and then sequenced. I got the material after it had already been sequenced, so I was just dealing with the raw sequencing reads. So once you start seeing the FastQC step here, that's where I came in. So FastQC, then I went into the mapping to the genome, counting reads associated with genes, and then of course, statistical analysis. So to dive into that a little bit more, this entire workflow, my project focused on the quality assessment of raw sequencing reads with FastQC and also multi-QC. I also completed alignment and gene count with star and salmon algorithms, which, like I said, would be these two steps here with the mapping to the genome and counting reads. And then after that, I identified specific genes and transcripts that are differentially expressed between samples and quantified expression levels of individual genes and transcripts. The resulting sets of differentially expressed genes can be further analyzed for the presence of relevant genes of interest, enrichment of functional gene categories, or overlap with sets of genes. I completed my project after going through this, this pipeline. I completed the project with hierarchical clustering, which can be used to discover characteristics shared in samples and predict function of genes. The goal of this project is to determine what genes are significantly expressed in samples. Specifically, I focused on genes expressed in racial groups classified as white and Black, and also genes expressed in the male and female subgroups for up to 500 participants. Cluster analysis was used to determine if there are any clinical characteristics shared by samples and to cluster genes to predict gene function. Ideally, data analysis will reveal significantly expressed genes that would indicate genetic differences between those who develop tics and those who do not. So a little background on the samples. As I said, this is RNA extracted from over 500 colon tissue samples. To elaborate a little bit on the library preparation method, that was mRNA stranded library preparation, and they were sequenced on the NovaSeq S2 platform. The library preparation and the sequencing was completed at the high throughput sequencing facility, which is a sequencing core on campus. And then, of course, after completed sequencing, that was when I took over with the raw sequencing reads. Another bit of information about these 500 participants, there were a few more women than men, so there is a bit of a bias there, and about half of the participants had tics and the other half did not have tics. So moving to the first step of the pipeline, the quality check. The FastQC provides a way to complete quality control checks on raw sequence data from high throughput sequencing pipelines. It also creates a set of analyses which can be used to provide an overview of your data and determine if it has any problems before doing further analysis. So here's an example of the code that I used. I completed this in Longleaf, which is, for those of you who don't know, it's a Linux-based computing system that is available to UNC researchers for things like data science and statistical computing workloads. So here, just to give everybody a breakdown, if you can see my cursor, we've got the amount of time dedicated to running the code, the memory that I was assuming I would use, the module, which in this case is FastQC, I'd load it here, and also where at the time the raw sequence reads were, and then also where I wanted them to be put at the time. This was a couple of months ago. All right, so then multi-QC. So the biggest difference between FastQC and multi-QC is that multi-QC takes the FastQC output and then provides a basic summary of everything. It aggregates all the information together. So it's a reporting tool that parses summary statistics from results and log files generated by other bioinformatics tools. And in the end, it gives a single HTML report file. So you can see lots of information on if there are duplicate reads, overrepresentation, if things need to be trimmed, etc. And here's an example of a multi-QC script that I did use. And you can see it follows the FastQC script very closely with the amount of time dedicated, memory, just a different module is listed here. And here's an example of the output that you can see once you run FastQC. So as I said, FastQC would be more of done on each individual sample, where multi-QC, you can see there are many different samples listed here. And you can also see we've got unique reads in blue and duplicate reads in black. So at the end of the day, when we were looking at all of this with my supervisor, we came to the conclusion that we needed to move forward with alignment and counting of reads. There were a couple of samples also. I don't think they're listed on this specific one, but they did not yield any reads. And so they were dropped from processing later on down the line, which I'll elaborate a little bit more on later. So as I said, I worked with my supervisor, and he wanted to align RNA-seq data with STAR, and then also use Salmon to count reads associated with genes. So STAR is an open source software that can be run on Unix, Linux, or Mac OS X systems. And it is a very fast and efficient tool. The alignment process includes choosing a reference genome to map reads against and performing the read alignment. And once again, I use this in Longleaf. And after aligning the sequencing output from all samples via STAR, STAR created a series of files which will be utilized by Salmon at the next stage of processing. So after I got the output files from STAR, I used Salmon to read those files, and Salmon is also a free software tool, fortunately, for estimating transcript level abundance from RNA-seq data. After reading in the files from STAR, it provided multiple different TSV format files listing the name of each transcript, its length, and its abundance in terms of raw count. After completing the count, all output files were merged in RStudio via the tax import library. The final output is a single file with the raw counts for each gene present in each sample. This file can then be used by statistical software such as RStudio with differential expression tools like DE-seq2 or EdgeR to analyze for gene expression, which is exactly what I did in the next step. So a basic task in the analysis of count data from RNA-seq is the detection of differentially expressed genes. The count data, which I described in the previous slide, are presented as a table, which reports for each sample the number of reads that have been assigned to a gene. The package DE-seq2 provides methods to test for differential expression and was completed in RStudio. And here's our different steps required for DE-seq2. So input data required for DE-seq2 include a table, the gene count file that I described previously, and also a metadata file, which you can see here. So the metadata file needs to have the demographic and clinical information on the subject, such as race, gender, and any other bits of information that you're interested in. And this file also included where the ticks were located and the presence and absence of ticks, which is going to be very important later when I describe the code that was used. So the count data file only showed results for approximately 480 samples. And as I stated previously, the metadata file had over 500. So I had to remove the samples that didn't make it in sequencing, that didn't yield any reads. And I did this in RStudio by selecting only the samples that were common in the count file and the metadata file. After removing samples with no reads, the results were further prepared by centering and scaling numeric variables in the metadata file, and we did this by the median. And to elaborate on why that was done, this was a massive project, like over 500 samples at first, and still 480 is quite large as well. So every single time I would try to run DE-seq2, my computer would crash, RStudio would crash, it would go incredibly slow. So in order to try to fix this, we were trying multiple different ideas to make it work. And finally, at the end of the day, centering and scaling the numeric variables, which in this case would be age and BMI, that was what finally allowed us to move forward with this project. So after the gene count file and the metadata file were prepared for processing, they were also renamed. So gene count would be base input, which you can see in this code here. And then metadata, the metadata file was base meta, which you can also see here. And then this information was loaded into the DE-seq object, which you can see in this entire figure below. By design, DE-seq2 will test the effect of BMI and age right here, because they're at the very end of this formula, while controlling the effect of binary tick here. So once again, that is the presence and absence of ticks. Here we can see one of the final volcano plots created with the DE-seq2 output. Volcano plots were created to visualize the results and identify genes with large fold changes that are also statistically significant. Upregulated genes are shown to the right, and downregulated genes are shown to the left side of the plot. The project was most interested in testing the effect of BMI, age, and sex on the presence of ticks and comparing the effect in both white and black populations. So that is why I've got this volcano plot here for the different racial groups. So black, and then this one, white. Now, those of you who are more familiar with volcano plots, you know that this is not quite what you expect from a volcano plot. Like typically there's a bit more going on, and especially for such a large project, this was not the expected result. There are far fewer genes that are significant than we expected. And due to this, moving to the next part, due to the unexpected nature of the volcano plots, an inner join was completed in R to determine which significant genes are present in both racial groups. However, only 83 genes were identified as being significant in both groups. Again, this shows far fewer significant genes than anticipated, especially for such a large study. I do have a sample of 5 that were present here. These are all protein coding genes. And for those of you who are interested, the value for significance was 0.05. So the last step of this project was to complete clustering analysis. Before I could do this, though, I had to change the raw gene counts file into a transcripts per million file, or a TPM file, which I did with this formula here at the bottom of the screen. And TPM is more popular right now when completing cluster analysis. It considers the gene length for normalization. After converting to TPM, I also filtered out low performing genes that were not expressed in at least 80% of samples. All right, so this, like I said, is the last stage of my project. Clustering, as many of you know, is a form of unsupervised learning. And through cluster analysis, we can group objects that are more similar to each other into a single group. The goal was to cluster samples to discover clinical characteristics shared by samples, or to cluster genes to predict the function of known and unknown genes. For this project, clustering was completed in Python using libraries such as Seaborn. We did try using a different software called Cluster 3.0 and then also Java Tree View, but unfortunately, again, this project is very large and my computer crashed quite a few times. Cluster 3.0 didn't like it either. We tried to use Cluster 3.0 on Longleaf and we did end up getting that to work, but then for some reason couldn't get Java Tree View to work. So we used Python and were able to get some output. However, it was not exactly what we expected either. So those of you who are familiar with hierarchical clustering will know that these significant streaks that go across the plot, that's just not exactly what you would anticipate seeing. So this was also very surprising. It shows that we do not see a lot of differential expression across samples. However, if you look at the dendrogram, you can see that there's clustering going on, but we don't know exactly what is going on to cause the groupings. So yes, like I said, not a lot of differential expression across samples. There are many clear clusters that are present across genes and across samples. But in conclusion, we don't know what is causing these clusters to group together. It is possible that there are different subtypes of diverticulitis. This is something that I spoke to my supervisor about. A lot of experts in the field have wondered if there are different subtypes, and maybe that could play a role in the unexpected output. Other conclusions from this is that the output at each stage of processing was just not consistent with the project's expectations. The volcano plots did not show a significant number of differentially expressed genes. The gene overlap also did not show as many genes as expected. Which brings me to future research that will be needed. Additional research will be needed to see if there are different diverticulitis subtypes that could be impacting these results. And also, future work will need to focus on processing the data in non-traditional ways to determine if that yields different output. Does anybody have any questions?